So I appreciate your attendance to talk a little bit about Lysogene today and what we're up to. So Lysogene is an advanced clinical stage gene therapy company. We are poised to be the first to develop and bring to commercialization a safe and efficacious gene therapy as a first-line treatment for MPS IIIA. Oh, I'm sorry. And we are also poised to develop a safe and effective gene therapy product for GM1. For the MPS IIIA program, and both of these programs I'll go into with some length. For the MPS IIIA program, which is our lead program, we are approaching a phase 2, phase 3 clinical trial planned for the first half of 2018, and that's an EU and US trial. And we plan our filings both in the EU and US. Those are expected in 2021, first half of 2021. So we are, I like to say, a dual headquartered company. It was founded, as I said, by Karen Ayesh, who is based in Paris. And 2 years ago, we opened the Cambridge office in Kendall Square. So we literally have half of our staff in Paris and the other half in Cambridge. And the benefit of that, obviously, is that we are, focused on both U.S. and EU, and that it serves the purpose well to focus on both of the environments and regions. So, as I said, our current focus is gene therapy. We are focused on central nervous system diseases. Here is the management team, which I'm proud to say is very experienced and qualified to run Lysogene, and even better still, the team behind this management team are exemplary. So we're very well situated to run this type of business and these types of programs. These individuals that you see here, average experience of 20 years within the field in their disciplines. So here's our pipeline within the CNS disease. As I mentioned before, we have MPS III. We're focused on MPS IIIA, and that program is LYS SAF 302. That is a second generation product, and that is currently entering into phase 2, phase 3. We have successfully concluded the phase 1 trial with 5-year data with a positive safety profile. And the GM1 program, which has just concluded basically proof of concept, and we're entering sort of the pre-IND stage of that, and I'll get into both of these a little bit more. From an IP and regulatory status perspective, we are well poised. There's actually an inaccuracy in this slide for LYS GM101, which is our second program. That actually hasn't been fully confirmed on the IP, but beyond that, the rest is accurate. We are well situated from a LYS SAF 302 for the MPS IIIA program from an IP perspective, as well as we have orphan drug designation for in the U.S., rare pediatric designation in the U.S., and orphan drug designation in the European Union for for both programs. So let me just jump into MPS IIIA a bit. So what is MPS IIIA? It's also known as Sanfilippo syndrome type A. It's an autosomal recessive lysosomal storage disease. The symptoms are the first clinical symptoms occur early in life. Severe behavioral, sleep, cognitive deficiencies are the key indicators of this, and prior to diagnosis, this is typically how they manifest before they are finally diagnosed. It quickly degenerates from there. Less than 15 patients survive beyond their teens. It is a very rare disease, one in 100,000 live births, and there is no approved cure. There is no cure at the moment. No real treatment for this disease. So, Lysogene's approach to MPS IIIA. It's understood that for MPS IIIA, SGSH enzyme correction, which is the enzyme in question, is critical in the CNS. And even with some symptomatic manifestations, this is clearly a neurological disease. The challenges persist in overcoming the blood-brain barrier to achieve widespread distribution in the brain where it is most needed. So for Lysogene, the clear mode of delivery was intracranial injection, direct injection into the parenchymal space. Once the drug is delivered directly where it's needed, the understanding is that the secreted SGSH enzyme can then have an effect on cells distant from the transfected cells via cross-correction. So now, as I said, we have 5-year data on the phase 1. We are very well situated to enter into the pivotal phase, and as part of that process, we learned a lot from the phase 1. We are optimizing the product. We've done a considerable amount of optimization for the what we call second generation product, that is LYS-SAF 302. And I'm going to just touch on a few of those at this point. So the first and most important is the potency of the product. As our original speaker, I forgot his name, but the first speaker of this session had made the comment that if you may have the right gene, but if you don't get it where it needs to be at the right amount, with the right potency, then you can stand on your head, as he said. And I agree with that 100%. So what we've done is we have optimized it from a potency perspective. We've changed the promoter from MPGK to a GAG promoter, which has improved the construct significantly from an enzyme activity perspective. We performed a MPS3A mouse model study, and you can see here, if I can figure down here, it doesn't work. You see the graph at the bottom, lower right. We performed a 4-week post-injection look at the MPS3A mice, as well as LYS-SAF 301, which is the first generation product, and then finally LYS-SAF 302. You can see here that LYS-SAF 302 did significantly better from an enzyme activity perspective when compared to MPS3A model, and also when compared to the first generation product. I would even argue that from a wild type perspective on the far left, it is, if you take into consideration that the margin of error, it is actually competing with actual wild type as well. So that's one element that was optimized. And then we also, started looking at the correlative nature of enzyme activity with regards to the endpoints for the trial. Most importantly, this is a secondary endpoint for us, is behavior, which is one of the key indicators of disease, as I mentioned before. For this study, we did a field test on mice, and the best thing to do is just run this, and you can see on the far right we have the mice, the wild type mice on the upper quadrant. We have MPS3A untreated in the middle, and MPS3A SAF 302 in the lower. And you can see that the SAF 302 did very well, and actually looks like it's very comparable to wild type, if not identical or better than wild type. We also wanted to evaluate distribution as an optimization element for the phase 2, phase 3. So we performed a dog study where we injected gadolinium in 2 injections per hemisphere, showing spread of, here you can see on the right, a very even spread front rostral to back caudal of the brain. And finally, in conjunction with that study, we also tried to optimize the device. So we're using a customized device for the phase 2, phase 3 trial, which will actually be our commercial device. We did a study with that to confirm that it was in fact appropriate. You can see here that with 1 injection per hemisphere, you have a very good distribution of the drug as analyzed by qPCR. So obviously with the arrow, you have the highest level, and then it spreads from there. And then what we did was extrapolate to the child model. For the pivotal study, this information is critical. So we took what we have from a large animal model and overlaid that into a typical child, 6-year-old child brain anatomy would be. We have determined that 3 injections per hemisphere are planned for the clinical trial, and that provides the coverage that we need, that we expect for efficacy. I see I'm running short on time, so I'll pick things up. We have completed a natural history study. The natural history study is incredibly valuable because there is, as I said, unmet need for this disease, and this will be used as the comparator for the phase 2, phase 3 trial. It will be combined with existing data that has already been published, and this will serve to be the comparator. It's important to note that on the right side of the screen, you see that we're using the cognitive Bailey assessment tool. And for this type of disease, a neurocognitive disease, it is very important to use the right tools to measure your endpoint. This has been widely recognized as the appropriate tool for, for this endpoint, and there are others that are being proposed or being used that are in fact less effective at analyzing data, especially in younger patients. We've checked all the boxes from a agency perspective. We've had several meetings with the FDA. We've had several meetings with the European Medicines Agency and the CAT, and we've had interactions with the PDCO with regards to our PIP for this. So we are well situated. We have agreement on our primary objective endpoints, sample size, first patient enrolled, and study duration. And finally, I'm just going to quickly cover GM1. As I said, is our earlier phase initiative at the moment. So GM1, it's also a lysosomal storage disease caused by mutation of the GLB1 gene, which encodes beta-gal enzyme. It manifests similarly in early infantile and late juvenile, and it has severe neurodegeneration, seizures, and it is exhibited with coarse facial features. It's 100% lethal. It's even more rare than MPS IIIA, and again, there's no approved or curative treatment for this. And we have performed a very, very promising study. It's hard to watch, but this is the best way to sort of, display the proof of concept that we have. We, in collaboration with Douglas Martin's lab in the Auburn School of Veterinary Medicine, there are GM1 cats. These GM1 cats were injected with the AAV-treated AAV gene therapy. And you can see on the right side, this is the cat that was treated. The one on the left obviously was not. The one on the left died several months after this video was taken. The one on the right is still alive and well. I think he's 7 years old. And he's apparently treated like a king, and he gets whatever he wants. So this is very promising proof of concept data. We are entering into the design of our clinical trial. We have developed a sort of adaptive study design, not just from a clinical perspective, but we have also have a very innovative design with regards to the manufacturing process, which I'll touch on next. In the interest of time, I'm just going to say 2 words about this, which is, we're holding one study, which will be a dose escalation study, and after 6 patients, following 12 months, we will initiate the efficacy study portion of that. We will obviously follow all those patients, but we do not intend to stop. We intend to use this one trial to get all of the data we need from a safety and efficacy standpoint. And then finally, I just wanted to also, as I intimated, that we have entered into a collaborative agreement with Brammer Bio. They are, a very good choice for a partner for this type of disease, for the manufacturer to the scale that we wanted to manufacture. As I said, we are streamlining, so we are entering into manufacturing from a basically high yield as opposed to incremental and then changing midway through and having to do comparability and revalidation. We're hoping to start the process the right way, and then that will be our clinical trial material as well as our commercial material. And I think that is my last slide. It is. I want to thank you all for hearing about Lysogene, and have a good rest of the day.